Imagine for a moment that cancer isn't just about rogue cells with, mutated genes. What if the very feel of a cell's environment, like how stiff or soft the tissue around it is, plays an equally critical role in how cancer behaves, how it grows, and even how it spreads?
It's a really profound thought, isn't it? It definitely broadens our view beyond just looking at the genetics, which has been the traditional focus.
Right.
And this deep dive, well, it's all about exploring that exact question. How does the stiffness of the extracellular matrix, the ECM, you can think of it as the scaffolding and goo around our cells, how does that directly influence the mechanics, the physical properties of cancer cells?
Okay, the mechanics.
And maybe even more surprisingly, how this physical environment impacts really fundamental things inside the cell, like how they package and move materials around.
So our mission today is to sort of uncover this crucial link, often overlooked, I think, between these mechanical signals and how cells behave, especially when we're talking about tumor progression.
Exactly, and we're mainly drawing on a really compelling research article that combines some serious data analysis with some pretty advanced biophysical modeling.
Gives us a few different angles on it then.
Precisely. A multifaceted look at this complex area.
Okay, so let's dig into this. What's the big clinical problem here?
Right, so the central issue, the kind of big question driving the research, is that tumor progression isn't just about genes going haywire. It's significantly pushed along by biomechanical signals from the surrounding tissue.
The physical push.
Yeah. When a tissue becomes cancerous, the ECM, that scaffolding, often gets dramatically remodeled, and typically it becomes much, much stiffer than healthy tissue.
Okay.
And this stiffness, it's not just a side effect, it seems to be a critical change that actively boosts oncogenic signaling.
Which basically means signals telling cancer cells to go wild.
Pretty much.
Yeah.
Grow, survive, invade more aggressively. And what's really striking, too, is that this mechanical stiffening also seems to affect how the tumor interacts with our immune system, sometimes even helping it hide.
Wow. Okay, so it's not just the genes. It's the physical crunchiness almost of the tumor's neighborhood dictating what happens next. That feels like a major shift.
It really is.
So how did the researchers actually investigate this? How do you measure the crunchiness effect?
Well, they used a pretty large data set from the NCI Leidos Mechanobiology project. They took 20 different cancer cell lines.
20. Okay, quite a few.
Yeah, a good variety. And they grew them on 7 different ECM substrates. So basically, they gave these cancer cells different physical environments, some really rigid, like glass, others much softer, using things like collagen and fibronectin gels at different stiffness levels.
They're testing how the cells react to different fields.
Exactly. And what they found was, well, it was really diverse. The cancer cells showed what they called a heterogeneous response, meaning there wasn't just one simple reaction across the board.
Okay, not a one-size-fits-all answer.
Not at all. For instance, the stiffness of the cells themselves varied hugely, not just between different cancer types like breast versus lung, but even within the same cancer type.
Right, within the same type. So 2 breast cancer cell lines could be very different mechanically.
That's right. Breast cancer lines actually showed a massive range in their stiffness. Some were quite soft, others surprisingly firm, even under the same environmental conditions, from about 3.5 kilopascals up to 11 kilopascals.
That's a big difference. So some cells are just inherently more, I guess, squishy, and others firmer.
Seems that way. And they saw patterns. Some specific cell lines, like certain skin or colon cancer cells, tended to have a lot of inherently stiff cells no matter what surface they put them on.
Okay.
But then conversely, many pancreatic, prostate, and lung cancer lines generally had more of the softer cells.
Interesting. But does the environment change them?
Ah, yes, and that's where it gets really interesting. Their sensitivity to the ECM stiffness also varied a lot. So while breast and colon cancer cells often got stiffer when they were put on stiffer collagen.
Which kind of makes sense, right? Stiffer outside, stiffer inside?
You'd think so. But some lung and pancreatic cancer cells, like one called PANC1, actually did the opposite. They showed a decrease in their own stiffness as the ECM got stiffer, both with collagen and fibronectin matrices.
Whoa, okay. That is counterintuitive. So a stiffer environment makes the cell softer.
For some types, yes. It just shows how uniquely different cell types have evolved to respond or adapt to their physical world.
That's a fantastic illustration. Definitely not straightforward. So, okay, if cells are sensing this external stiffness and changing their own stiffness, the next question has to be, How? How are they doing that internally?
Right. What's the mechanism inside the cell? And that brings us squarely to the cytoskeleton.
The cell's internal scaffolding.
Exactly. It's absolutely key. So the researchers dug into this using some pretty sophisticated tools, machine learning, information theory applied to huge amounts of multi-omics data.
Genomics, proteomics, all the omics.
Yeah, all the omics, yeah. From 9 of the cell lines in that NCI Leidos dataset. They were basically hunting for a kind of molecular fingerprint that directly correlated with these changes in cell stiffness.
And did they find one? Like a master control panel for stiffness?
They found something pretty close. They identified a set of 18 specific proteins, all related to the cytoskeleton, whose expression levels correlated really strongly with how stiff the cell was.
18 proteins, okay.
Yeah, things you might expect, like actin, which forms microfilaments, intermediate filaments like vimentin and keratin and lamin, microtubules made of tubulin.
The main structural component.
The main building blocks, exactly. But also crucial accessory proteins, things like alpha-actinin, filamin, plectin, talin, vinculin, myosin. These are proteins that help organize the scaffolding, bundle filaments, link them to the membrane, and generate contractile forces.
So these 18 proteins are kind of like the cell's internal architects and engineers, letting it adjust its structure and tension.
That's a great way to put it. And the power of this finding was pretty striking. They built a neural network model using only the expression levels of these 18 proteins.
Okay.
And it could predict with about 98% accuracy whether a cell would fall into a low or high stiffness category.
Wow, 98% just from those 18. That's incredibly predictive.
It really is. It strongly suggests a direct, measurable link between expressing these specific proteins and the cell's physical state. And they went even further, looking at the top 5 most predictive proteins using something called Shapley analysis.
Which ones were they?
They were alpha-actinin 4, ACTN4, myosin 9, MYH9, plectin, PLEC, talin 1, TLN1, and vinculin, VCL. And what they found was that higher levels of ACTN4 and PLEC pointed towards increased overall internal stiffness, like more rigid scaffolding.
Okay.
Whereas higher MYH9 suggested increased tension right under the cell surface, in the cortex, like pulling a drawstring tighter around the edge of the cell.
So it's distinguishing between overall rigidity and that sort of surface tension.
Exactly, and this directly links what's happening outside the ECM stiffness to the specific types of internal forces and structures within the cell.
That's a really deep connection, linking the outside world to the inner machinery. But, the crucial question always is, what does this mean for actual patients? Does this lab finding translate to real-world outcomes?
Absolutely vital question. And this is where the research takes that critical step from the lab bench towards clinical relevance. They performed survival analysis using a huge public database, the TCGA Pan-Cancer Atlas, accessed via CBio portal. This has loads of clinical data from thousands of patients across many cancer types.
Okay, so they're looking at real patient data now.
Yes. Specifically, they checked if the expression levels of those key cytoskeletal genes they identified correlated with patient prognosis, essentially. How long patients survived or how their disease progressed.
Did they? Is there a connection?
There is. They found that the expression levels of these specific genes do correlate with patient prognosis across several different tumor types. They looked at ovarian, skin, brain, lung cancers, among others.
Okay, that's significant.
But the effect wasn't uniform. It wasn't like high expression is always bad or low expression is always good. Once again, it really highlights the complexity and the specific nature of different cancers.
Right. Can you give us some examples? How did it vary?
Sure. So, for instance, in skin cutaneous melanoma, higher expression of some of those key genes we mentioned, like ACTN4, MYH9, PLEC, actually correlated with lower median survival.
Okay, so in skin cancer, having more of those tension and stiffness components seem to be linked to a worse outcome.
For those specific genes in that specific cancer, yes. But then if you look at, say, ovarian cancer or lung adenocarcinoma, in some cases, it was actually low expression of a particular cytoskeletal gene that correlated with lower median survival.
So the opposite effect.
In some instances, yes. And to really drive home the complexity, take one specific gene, LMNA, which codes for lamina AC, part of the nuclear envelope structure. Low expression of LMNA was linked to lower survival in ovarian cancer patients. But surprisingly, low LMNA expression was linked to higher overall survival in brain cancer patients.
Wow, so the same gene same low expression, has opposite correlations depending on the type of cancer.
Exactly. It really underscores that context is everything. The tissue type, the specific cancer subtype, you can't just draw simple conclusions across the board.
That's a critical point. It's deeply personalized, deeply context-dependent. Okay, so we've traced this from the outside environment's stiffness to the cell stiffness, down to the specific proteins involved, and now even link their expression to patient survival, albeit in complex ways. Let's go even deeper now. How does all this connect to something even more fundamental, like how cells move things around internally or communicate? This feels like where the really profound mechanism might lie.
I agree. This is where we get to some core cell biology, the really fundamental insight. We need to talk about trafficking vesicles.
It's like the little sacs that move stuff around inside cells.
Exactly. Cells constantly form and move these tiny sacs. They're absolutely essential for communication between cells and for all sorts of processes within cells. And unfortunately, these pathways are often, well, hijacked by cancer.
Hijacked how?
Well, think about exosomes. These are nano-sized vesicles that cells release to communicate with each other. In cancer, tumor cells release exosomes that are packed with signals that can promote invasion, help the cancer migrate, and even suppress the immune response. They're like little messengers carrying bad news for the body.
So the cancer is using the cell's own delivery system against it.
Precisely. And forming these vesicles, these exosomes or others involved in internal trafficking, requires the cell membrane to bend quite dramatically, to curve and butt off.
Right, it has to physically change shape.
It does. So the study asked a really fundamental biophysical question. Does the stiffness of the ECM, by affecting that internal cortical tension we talked about.
Drawstring tension.
Exactly. Does that tension then affect the cell's ability to generate the membrane curvature needed to make these trafficking vesicles?
Okay, that's a cool question. Connecting the outside stiffness all the way down to the formation of these tiny internal packages, what did the models show?
They showed a clear yes, and not just yes, but with what they described as an ultrasensitive response.
Ultrasensitive?
Yeah.
Meaning a small change has a big effect.
Exactly that. Their mechanistic biophysical model showed that the membrane deformation, the bending caused by the proteins that actually induce this curvature, is ultrasensitively dependent on that cortical tension. It follows something called a Hill-type function, which basically means you hit a certain threshold and bam, you get a huge change in response for just a small further change in the input, in this case, tension.
Like a switch flipping almost.
Sort of, or like a very finely tuned amplifier. A slight adjustment in tension can lead to a disproportionately large change in how readily vesicles or tubules form.
Okay, so if the internal tension in a cancer cell is, say, lower, does that make it easier to pop off these vesicles? Could they churn them out faster?
That's precisely what the model suggests. Lower cortical tension makes it easier for those curvature-inducing proteins to cluster together and do their job, promoting the formation of highly curved structures like vesicles.
Okay.
Conversely, the model also showed that if a cell manages to get more of these curvature-inducing proteins onto its membrane, it can still form protrusions even if the tension is higher.
So there's a balance between the tension and the proteins doing the bending.
Right, and what's really key here is that this fundamental tension-dependent way of forming vesicles, combined with the idea that the membrane tension also seems to affect how many of these bending proteins get recruited to the membrane in the first place.
So tension affects both the recruitment and the action of the proteins.
Exactly. This suggests what the researchers call a powerful mechanosensitive regulatory axis. It implies that cancer cells could potentially manipulate this whole system, tweaking their tension, altering protein recruitment to hijack intracellular trafficking for their own benefit, survival, spread, communication.
That's just incredible. A fundamental physical control switch that cancer might be exploiting. So let's try and weave this all together. We started with the stiff outside environment affecting the cell's own stiffness.
Then we found specific cytoskeletal proteins mediating that chain,
which in turn affects the internal cortical tension.
And that tension has this ultra-sensitive impact on how easily the cell membrane can bend to form vesicles.
It really paints a picture, a chain reaction from the physical environment right down to basic cellular machinery, potentially explaining another way tumors progress.
It does. It creates what the paper calls an intriguing paradigm of mechanosensitivity influencing core cellular functions. It gives us a more holistic view, We already knew, for example, that messing with trafficking pathways is bad in cancer.
Right. Things like changing how cells recycle surface proteins involved in invasion, or how they get rid of growth signals.
Exactly. Enhanced recycling of integrins helps cells invade. Disrupted degradation of growth factor receptors keeps proliferation signals switched on. We knew those things promote cancer. This research adds this crucial physical layer to that picture, showing how ECM stiffness and the resulting cortical tension can directly influence that very trafficking machinery.
And you mentioned earlier there's a genetic angle to this trafficking machinery too, right? It's not just the mechanics being manipulated.
Absolutely. The researchers also looked at genomic data, again, from CBioPortal patient samples, and they found that key protein families that regulate this whole vesicle trafficking system, the organizers and controllers, show significant alterations in cancer.
Like which ones?
Things like RAB-GT passes, they're like molecular switches controlling vesicle movement. Sorting nexins, SNXs, which help sort cargo. Components of the exosomes complex involved in docking of vesicles at the membrane, and autophagy-related proteins, ATGs. These families frequently have mutations or copy number variations, bits of the genome duplicated or deleted in patients across many different tumor types.
So it's like a double whammy again. The physical environment is pushing the system, and the genetic controls for that system are often broken in cancer as well.
Precisely. It really reinforces the clinical importance of these trafficking pathways, governed by both mechanical cues and genetic integrity.
So thinking ahead, what does this open up? Where does research go from here, and what could it mean for maybe new cancer treatments down the line?
Well, the potential is really significant, I think. The next steps would be to try and integrate these complex signaling pathways, the ones triggered by the mechanical cues from the ECM, with the detailed biophysical model of vesicle formation.
To get an even clearer picture of how it all connects.
Exactly. A deeper understanding of precisely how tumors manage to deregulate vesicle biogenesis and trafficking for their own ends. And if we can really understand those mechanisms, well, that might uncover entirely new types of druggable target.
Targets that aren't just the usual mutated genes.
Right. Targets that focus on the mechanical properties, the structural aspects of the cancer cells, and their interaction with the environment. Maybe ways to interfere with how they sense stiffness, or how they regulate their internal tension, or how that tension controls trafficking.
So stepping back, the big takeaway is that the physical world of a tumor, its stiffness, its structure, profoundly influences how cancer cells behave. It does this through a really complex interplay of cell mechanics, specific proteins, internal tension, and even the basic machinery that moves things around inside and between cells.
That sums it up beautifully. It's a physical dimension to cancer that we're really only beginning to fully appreciate.
Which leaves us with a pretty provocative thought for you, the listener. Imagine a future where we don't just fight cancer with chemotherapy or drugs targeting specific genes. What if we could also fight it by subtly altering the physical stiffness of the tumor environment itself, or by precisely tuning the internal tension within the cancer cells? How might a mechanical approach like that revolutionize cancer treatment?
It could truly be transformative, adding a whole new set of tools to our toolkit, moving beyond just the chemical and genetic into the physical realm.
Something definitely worth thinking about: the hidden physical forces shaping life and disease, and how understanding them might just unlock completely new ways to heal.